second presentation of the night is Emerging Therapies for Hemophilia. Dr. Stephen Pipe has joined us this evening to share his expertise. Dr. Pipe is a professor of pediatrics and pathology at the University of Michigan, the pediatric medical director for the hemophilia program, and has been involved in clinical trials in recent years with novel therapies for hemophilia, including gene therapy. Thank you for being here, Dr. Pipe.
Well, thank you, and I'm really pleased to be here, and I'm excited to give this presentation, and especially following Tommy's talk there. I think he really set a high bar for how to communicate a really complex subject like gene therapy and really make it accessible for everybody. And so that allows me now to talk about some other novel therapies that I think are important. They are emerging, meaning that they're still under investigation. But we have the first product that's in this category that I'll tell you more about as well, that's already in common use. So what I'm going to review with you tonight, I'm actually changed my mind a little bit. I want to spend enough time on some of these novel therapies and their mechanism of action, because I don't think they get as much attention as gene therapies, since gene therapies are well down the line of investigation here. So, I mean, if we have time at the end and people have some comments about just general risks and benefits of gene therapy, we can certainly do that in the Q&A. But I want to dive into some of these other therapies that I don't think you've heard that much about before. And I think to set up why we're even doing investigations in all these different types of therapies, we need to understand what we currently have at hand, what their limitations are, and why we might need some out-of-the-box thinking to take us to new levels of care for patients. So this is a new theme that I think has been evolving over recent years, and this is the idea of focusing on patient-relevant outcomes. A lot of clinical trials and innovations focus on the clinical outcomes. Clinical outcomes are things like joint bleeding, pain, how you manage in response to a treatment, a factor level. Those are all sort of clinical outcomes. But what we really should be focusing are patient-relevant outcomes. And this is along a theme that has been talked about under health equity. And the idea is that we should be developing therapies and modalities of care which allow people to ultimately climb the stairway of health equity towards having optimized health and well-being. And in order to achieve that, we probably have to move the clotting system so it's essentially normalized with our treatments. Now, if you look at this stairway, you'll recognize that it wasn't that long ago that we were talking about therapies that were just designed to prevent premature death, improving quality of life by allowing people to just participate in the normal activities of daily living. So that was sort of the state, if we go back to the late 60s and early 70s. During the last, 4 decades or so, we've really made tremendous advances with the clotting factor treatments and their availability. And now I think as we've continued to optimize prophylaxis particularly, you can see that we've made several additional advances on this stairway. We have patients now that can be free from at least spontaneous bleeding. They can attain normal mobility. In some cases, you can even sustain minor trauma without needing additional treatment. People are participating in work, career, and family life without restriction. And if I think about some of our younger adolescents and younger men in our clinics, they seem to be living a fairly unrestricted lifestyle with the availability of the therapies we have today. But if we look at those last couple of steps, it's going to be increasingly challenging with the tools that we have in hand to get to the place where patients can undergo surgery or have major trauma and not need any additional intervention. We really haven't normalized anybody's blood clotting system. And so if to get these last couple of steps, we're going to have to continue to look to new innovations. Now, we can't talk about health equity without talking about the complication of inhibitors. And as we talk about new therapies, we don't want to have a situation where it seems like people with inhibitors are being left behind. So what I'm really focusing on tonight actually is to give you some insight that some of these emerging therapies are uniquely positioned so that they will be effective in both patients without inhibitors as well as with inhibitors. And the reason we can't let patients who've developed inhibitors to factor VIII or factor IX be left behind is because of the significant consequences. This is primarily a problem of people with hemophilia A because it occurs in about 30% of those with severe hemophilia A, but it's a reality for some people with severe hemophilia B, and in some cases, those who develop inhibitors with hemophilia B, this is even a bigger challenge because the immune tolerance regimens don't work as well in hemophilia B. And uniquely, this population has a preponderance of developing anaphylactic responses to factor IX coincident with inhibitor development that even precludes being able to do tolerization. So when we think about inhibitor challenges, these patients can have uncontrolled bleeding, have more chronic progressive joint damage. Their overall treatment burden is higher. They have higher mortality and hospitalization rates. There are psychosocial challenges. Surgeries can certainly be challenging, and overall treatment costs are significantly higher. So when we're talking about these new therapies, we want to keep in mind that we're looking for therapies that are also going to advance health equity for those who have been challenged in their care due to inhibitors. So I've used this slide for a long time to sort of walk you through some of the eras of treatment modalities. Now, I mentioned sort of the pre-replacement therapy era goes back to really, the early to mid 1960s. The replacement therapy began that era with the ability to use plasma-derived clotting factors and purify them from donated plasma. Initially as precious resources, these were reserved on demand to treat acute bleeding. But even back then, we recognized the benefits of prophylaxis, and that if patients could be given these clotting factors prophylactically, we could convert their severe phenotype or the expression of their disease to a milder version. Now, we had all kinds of plasma-derived components to treat various inherited bleeding disorders, but it was the recombinant platform that's really allowed us to develop some additional innovations. So similar to what Tommy walked you through with the sort of providing a good copy of the gene into the liver cell, what we do is we take a good copy of the factor VIII or factor IX gene, we put that in cells, and we have the cells make the factor VIII or factor IX. But once we have that platform in place, we can do some other things, and we can start to modify those molecules, and we can add things to them that change some of their properties. We can make them bigger or smaller. We can enhance their activity and make them more potent. And so when we talk about bioengineered variants or bioengineered recombinant factors, these are modified at the genetic level and then produced in the cell lines. And the most successful bioengineering has actually been to extend the half-life of these replacement therapies. And we'll talk about that in a few minutes, how those have helped, and then where else we can go with that technology. Replacement therapy probably takes us a good way of the journey in that health equity stairway I showed you. What about this new category that is called non-replacement therapies? So these are drugs which, instead of replacing the missing factor VIII or factor IX molecule, are acting on blood clotting in some different ways. And there are two broad categories that I think we can think of in this non-replacement therapy group. The first one is to use a substitute for factor VIII. This uses a molecule that acts as what's called a mimetic. Some people might call it an agonist. What this is this is a bispecific antibody. Antibodies recognize proteins. And it's a bispecific antibody that recognizes activated factor 9 and factor 10. And it turns out that these are the two molecules that factor 8 interacts with in the clotting cascade. So this antibody can mimic the action of factor 8. Factor 8 binds on the platelet surface, and it brings factor 9 and factor 10 together. And what this bispecific antibody does is it mimics that activity. And the beauty of this molecule is it looks nothing like factor 8, which means anybody who has an inhibitor to factor 8, this molecule will not be neutralized by that antibody. And so this is the first therapy that allows effective substitution therapy in an inhibitor as well as a non-inhibitor context. Now, the other broad category, and these are all in investigation currently, these are looking at something called hemostatic rebalancing, and I'll get to that in a few minutes. And so what we're doing there is we are attacking the other side of the coagulation balance, and instead of replacing what's missing, we're knocking down the function or the levels of the proteins that are trying to counteract those procoagulants like factor 8 or factor 9. And then we've already heard about gene therapy and what's going on in that area. So this is another, I think, important slide to help you understand what's different about these different modalities of therapy. So what's depicted across the bottom are measurable factor levels with these different therapies. So if you are using factor 8 or factor 9, you give yourself an infusion and you immediately raise your level off your baseline based on the dose that you give up to usually quite high levels. Most people are doing prophy up anywhere between 50 and 100% correction. But because the half-life of these agents are pretty short, your half-life of the molecule determines that you're going to lose those plasma levels pretty quickly. And particularly for factor 8, you're right back down to your baseline within about 48 to 72 hours. And so then you have to infuse again. And so you get this sawtooth pattern to your replacement therapy. What about the extended half-life factors, these bioengineered molecules? Well, they do a couple of things. They do last longer in circulation. They still have this peak right after infusion, and then they lose activity over time, but it's slower. And if you dose before you get all the way back to your previous baseline, what it does is it raises what we call the trough level. And we think this trough level is important in staving off particularly spontaneous bleeds. And so what we've seen with the extended half-life factors that have been available the last 5-6 years is that patients have a more optimized prophylaxis. They have a raised baseline troughs, better protection from spontaneous bleeding, and the overall burden of treatment is less. And these factor levels are mirrored, if you like, by the hemostatic potential or the clot potential of those factor levels. And so you get a sawtooth pattern of regular factor and this pattern with the extended half-life factors. Now, I mentioned that substitution therapy. Of course, this is emicizumab or Hemlibra. This is the substitution therapy, the bispecific antibody for factor 8. Because it's not factor 8, it doesn't produce any measurable factor 8 level. So now we have this interesting situation where we don't have a measurable factor 8 level, but we do have a hemostatic effect. And more so, the difference of this therapy is it's a steady state level. So what happens when you use emicizumab is you do a loading over about 4 weeks. And then once you get up to your peak level, then you continue with a maintenance dosing on it, either weekly, every 2 weeks, or every 4 weeks, which I'll show you in a minute. And what that does is it takes you up to what we call a steady state level. And here, the hemostatic potential no longer is the sawtooth patterns, but a steady state, even correction of blood clotting. Now, it's subnormal. So this is not normalization of hemostasis, but it does move you up into levels which are protective, at least against spontaneous bleeding, and for most people protective against most joint bleeds. Gene therapy is also unique in that once you start expressing from that liver cell, as Tommy walked us through, you're going to reach a steady state level of measurable factor VIII or factor IX in the bloodstream. And this is represented here. So 24/7, 365, you're roughly going to have the same factor level all the time in your blood. And that's going to have a corresponding hemostatic effect to protect from bleeding. And how high that level is going to determine how well you're protected from that gene therapy. Now, to give us a little bit of additional insights on what's going on with some of these new developments, this is a representation of the risk of having joint bleeds in a particular year correlated against whatever your residual factor VIII activity level is. So if you have severe hemophilia, you have no factor VIII or factor IX. And so you have less than 1% activity. And based on some natural history study of people with hemophilia A, those who have severe hemophilia have the highest risk of having joint bleeds in any given year. But even having 1 to 3% residual factor VIII, what we would call the moderate range of hemophilia, has a significant impact in reducing the risk of joint bleeds. And if you get above 5%, which is now the mild hemophilia range, joint bleeding is pretty unusual. And above a level of somewhere around 12 to 15%, we generally don't see joint bleeds occurring, even in the context of mild hemophilia. Now, the traditional prophylaxis I talked to you about was aiming to keep trough levels so that they didn't drop below 1%. And I would say for about 2 decades, that's the way prophylaxis was performed. And we recognize that we can optimize things further, including using what are called pharmacokinetic profiling of patients, so actually measuring their levels after dosing to optimize their dose and their interval, and also leveraging the bioengineered extended half-life molecules. This can now get us up into a more optimized prophylaxis with better outcomes. To get into this degree of protection in this area, we've had to use new therapies. And these new therapies have been compared against traditional and optimized prophylaxis and proven to have superior bleed control. So many would argue that the new standard of care is to get people's level of correction into this area. Now, aspirationally, we would love patients to be even further towards the normal range, and we're definitely going to need new technologies to get us there. We hope that gene therapy will do that for a lot of patients. Now, if we have to rely on replacement factors to accomplish this, the burden of the treatment, the administration, the frequency of the infusions, the cost of the infusions, this only gets higher as we keep pushing towards a higher and higher standard of care prophylaxis. And so that becomes challenging, and it only takes us so far. And what I'm also going to show you is that there is a ceiling for these so-called non-factor therapies. They don't seem to be able to carry us all the way towards normalization. And so there's a partial correction of the blood clotting, but then it sort of caps at a ceiling, and that's a limitation for these therapies. Now, we're going to use this target to sort of give you a sense of all the activity that's going on in these areas. So the center in green is the target. That's an approved therapy by the regulator, so the FDA, and it's marketed and available for patients. The only one of the therapies I'm going to talk to you about today is emicizumab that has made it all the way this far. But you can see that there's a number of therapies that are in what are called the late phase 3 portion of the clinical development programs. And so these are all either being reviewed or imminently will be completing their clinical trial programs and submitting for review with the regulators. And these include some additional gene therapies, as well as some of the novel therapies that I'm going to talk to you about today. And then on the periphery, we see some early phase activity, again, some gene therapies, as well as some new non-factor therapies that are either about to enter the clinic or have just entered the clinic that I think also offer some promise. And we'll talk to you more about that in a minute. Now, what I want to point out for you here is look at the shapes that are chosen for this graph. Some of these are only designed for hemophilia A without inhibitors. This is going to be your gene therapies. Same thing, hemophilia B with inhibitors. These are going to be gene therapies because we can't use gene therapy as it is today to treat patients who have inhibitors. But if we look at those that are in the triangles or in the circles, we now are seeing some cross-platform efficacy or effectiveness of these therapies, whether it's hemophilia A with and without inhibitors. So emicizumab's the best example of that. But now we have some therapies that are going to work for hemophilia A, hemophilia B, with and without inhibitors. So cross-platform across all of the hemophilias, whether you haven't had an inhibitor or not, these therapies offer an opportunity to have effective prophylaxis. So let me remind you a little bit more about what we've seen with emicizumab. This is the first in the category of these non-factor therapies. So what I described to you before is factor VIII's function. It sort of acts like I like to think of it as a scaffold. So factor VIII settles on the activated surface of the platelet, we call the phospholipid membrane, and then it allows the assembly of factor IX and factor X to come together into this complex. And by doing so, it helps factor IX get oriented so it can efficiently convert factor X to 10A and blood clotting can move forward. That's normal physiology. If you're replacing factor VIII with an infusion, the limitation here is that factor is only going to last in circulation probably about 40 hours or so, maybe 48 hours, because the half-life is only about 6 to 10 hours. And that's why we see the sort of sawtooth pattern that we have here. Emicizumab, as a bispecific antibody, has several additional properties which give it an advantage. It mimics the function of factor VIII, because one arm of the antibody recognizes factor IXA and the other arm recognizes factor X, and it brings them together and allows factor IXA to activate factor Xa efficiently. But the other properties of this molecule are impressive. First of all, antibodies can be absorbed readily when they're given under the skin, and you only need a small volume of these because they're highly potent. And so you can give this subcutaneously instead of IV infusion. And then look at the difference in the half-life, 6 to 10 hours versus 28 to 30 days. So here's that loading phase I described to you. 4 weeks of loading gets you up to the target level. And then once you stay on a maintenance regimen, because of this really long half-life, you will stay at a steady state plasma level, and this corresponds to the hemostatic effect of the molecule. Now, this has been examined in both hemophilia A with and without inhibitors. It's been examined initially in adolescents and adults in HAVEN 1. Then it was studied in HAVEN 2 in pediatric patients with inhibitors. Then it was studied in non-inhibitor adolescents and adults. That was the HAVEN 3 study. It was then explored in adolescents and adults with a very extended regimen of every 4 weeks. And since this time, we've done 2 other studies. HAVEN 6 was a study in non-severe hemophilia, so mild and moderate hemophilia. And then what we're actively engaged, we're one of the centers studying this in essentially infants in the first year of life. And so they are offered to initiate on emicizumab shortly after birth, all the way up through the first year of life. And then this becomes their prophylactic therapy. So the different regimens that have been part of the studies are once a week dosing, every 2-week dosing, or every 4-week dosing. So it doesn't matter which regimen you choose. You might say, well, is the 4-week, does that mean I'm going to have more breakthrough bleeding? That has not been demonstrated in the trials. And it turns out that they all get loaded similarly. And once they're up in this band, this wide band of levels, everybody seems to have the same hemostatic protection. So whether you're on weekly dosing every 2 weeks or every 4 weeks, this maintains an even protection against bleeding. And it doesn't seem to matter whether you're adult or a child. The levels are also in this protective band. So that's allowed us to do similar dosing in the pediatric patients. And what we'll be showing at a big hematology meeting in just a few weeks, we're going to be presenting the data from the infants on Haven 7. And nicely in that study, the infants also achieve very good levels that will allow them to have effective prophylaxis. So is the level of protection that's provided with emicizumab superior to clotting factor concentrates? Well, this has now been demonstrated in 2 of the trial programs I mentioned to you. In Haven 3, which was the non-inhibitor patients, looking at Q weekly or every 2-week dosing. They looked at a number of bleeding endpoints, and the punchline is there was a 68% reduction in treated bleeds and a 62% reduction in treated joint bleeds, and a higher proportion of patients who experienced 0 bleeds in the follow-up compared to their prior factor VIII prophylaxis. This was the first new therapy since clotting factor concentrates came out to demonstrate evidence of superiority over traditional factor VIII prophylaxis. And most notably, if you're familiar with target joints, these are joints, usually ankles, knees, or elbows, where patients are bleeding, 3 times or more within a 6-month period. And patients who came into the trial with target joints, essentially all of their target joint bleeding was eliminated. And then in Haven 4, this confirmed the similar endpoints observed here with the every 4-week dosing, also demonstrating whether you had inhibitors or not, the 4-week dosing was effective. And this is showing the pooled, what are called annualized bleed rates. The patients all had improvements in their annualized bleed rates compared to their, how they came into the trial. And then they got down to very low levels and stayed down there, essentially at levels that are approaching, 0 to 1 bleed over the course of a year, extrapolated. And as long as they stayed on the maintenance therapy on emicizumab, they had excellent control. And this is also, I think, important data here. This is looking at the proportion of patients who experience 0 treated bleeds from the first year and beyond. And you can see that over the first year after starting emicizumab, about 80% of them are having 0 treated bleeds. And that stays up at that level or even gets higher when they're followed for subsequent 24-week intervals. Now, I want you to think about that sort of optimized prophylaxis we talked about at the beginning. How much protection is emicizumab providing as the first example of a factor VIII mimetic? I told you it's not factor VIII, so we can't measure a factor VIII levels, but there are strategies to sort of come up with some estimate of an approximate level of hemostatic protection and comparing that to a factor VIII level. And most people would accept that emicizumab at that steady state level I showed you takes you up to an equivalence of maybe in the range of 10 to 20%. Well, what does that look like related to risk of bleeding? If you need surgery, I'm going to want you to have your factor VIII level boosted to well above 50% and really approaching 100%. When you're recovering from surgery, we can maybe let that drift a little bit lower, maybe below 50%, but definitely in sort of the 30 to 50% range. If you want to perform some pretty intensive physical activity, playing in an athletic sport, going on an aggressive hike, downhill skiing, anything along those lines, you probably need a factor level that's at minimum in this range. If you have a job where you're on your feet all the time or you're walking all the time, etc., you probably need a factor level that's much higher than the trough levels that are typically done for prophylaxis. So when we think about this degree of hemostatic protection, we're kind of down in this range. And so the issue is, do the factor VIII mimetics, emicizumab, the non-factor therapies have been examined so far, is that going to allow patients to do whatever they want to do without risk of bleeding? The answer is no. Are they going to be able to have major surgery and not need any factor products? The answer is no. So that's that ceiling effect from the non-factor therapies that we still have to deal with. And so we're looking for new technologies that might give us some additional options. Well, is emicizumab the only example of this mimetic strategy? Well, there's actually another molecule that's being looked at. This is the one that's farthest along in the clinical trials. It's called Mymate. It is a similar mechanism of action of emicizumab, so it's a bispecific antibody that recognizes factor IX and factor X. But what they've done is they've tweaked this antibody, so they've altered the affinity, and they've made it so essentially it's more potent. And so we've seen the first data from a conference this summer. And looking at observations in patients, what we can see is that this is a measure of what's called the thrombin potential, or how much thrombin is generated in a test tube once a sample is taken from a patient. And higher is better. And what Mimate allows you to do is generate more thrombin with a lot less drug. So it's clearly showing its potency. What that's potentially going to allow is you would be able to use a lot lower volume of this molecule to do effective prophylaxis, but it also may be able to take patients to a higher level of hemostatic protection. So as the clinical trial moves forward, we can look forward to see whether Mimate really, or Mymate, delivers on this. There's at least one other bispecific antibody. It's made by the originators of the emicizumab molecule. This is called NXT007, and this is also a potent molecule, perhaps has even higher potency than this Mymate molecule does, but that is just about to enter clinical trials now. So what are we looking for from the non-factor therapies? What's going to take us to sort of the next level of protection? So these are some of the unmet needs with the existing therapies. We still have breakthrough bleeds, no matter what therapy is used, clotting factor concentrates, even with emicizumab. Our goal is to get to 0 bleeds in all patients, particularly joint bleeds. We would like to reduce the treatment burden overall. We want to have easier and less frequent administrations. Emicizumab has definitely delivered on that. We would like to either avoid developing inhibitors, or if we did develop inhibitors, we would like it to not have so much consequence for the outcome for a patient. So if we have treatments that are available for persons with hemophilia, both with and without inhibitors, that's a great advance. We want to continue that march of up the stairs of health equity. We want to improve the quality of life in patients. And one of the things that they're looking for is to be able to live active lives without having to feel like they're at risk for breakthrough bleeding. And many of them want to live similarly to their non-hemophilic peers. And then the other thing, we want to make sure that we have access to treatment. Health equity isn't achieved if it's only accessible to a limited number of patients. So having therapies that are accessible to all persons with hemophilia. So let me talk to you about what may seem like a traditional treatment, but it's really an elegant innovation in the bioengineering category. So some of you might be familiar with the extended half-life factor VIII molecules. FC fusion, this is lactate. N8GP is a glycopegylated version of factor VIII that's made by Novo. And then you might be familiar with Jivi, which is also a pegylated molecule of factor VIII. And then this is Adinavate, which is, so this is Esperoct, and this is Adinavate, which is also a pegylated molecule. These are all bioengineered factor VIIIs trying to enhance and prolong their half-life. And what I want to show you here is their half-lives are all very similar, anywhere from 14 to say 19 hours. Now that's longer than the 6 to 10 hours I showed you a few minutes ago. So they definitely are extended half-life molecules, but they are only modestly longer than the traditional standard half-life agents. And the reason for this is you may recall that factor VIII's primary partner in circulation is von Willebrand factor. And von Willebrand factor binds to the factor VIII molecule, stabilizes in the plasma, and also what that does is it blocks factor VIII from degradation or from clearance from the plasma, but it also locks factor VIII for clearance along with von Willebrand factor from the plasma. So what it does is it creates a ceiling for how long the half-life of factor VIII. So basically what all of these extended half-life bioengineered molecules are running up against is they can't have a half-life that's any longer than the half-life of von Willebrand factor. And so that limits how long we can really make the molecule. The only way we can, so far, we can extend this half-life even further is we have to divorce factor VIII from its partner, from von Willebrand factor. So let me show you how that's done. This molecule is called BIV01, initially during the clinical development program. It has a number of innovations which allow it to be stabilized in the plasma and divorced from von Willebrand factor. And what the primary thing they did is they took a recombinant fragment. So remember, this is fragments that are made in cells. And this happens to be a copy of the fragment of von Willebrand factor that naturally binds and stabilizes factor VIII. So it doesn't have the rest of the von Willebrand molecule, just this, what's called the D' D3 portion. And what they do is they link it with the FC fusion. So this is a lactate. This is the FC fusion molecule of factor VIII. And then what they do is they link these together so that this D' D3 shields the factor VIII and protects it in circulation. And then on top of that, they ended these squiggly inserts. These are sequences of amino acids, so like protein elements. And what they do is they change the characteristics of the molecule, so it buffers it away from clearance receptors. So these are called X10 insertions. So both of these combined generate a molecule that has a very long half-life. Now you would say, well, how does this still function as a factor VIII molecule? Well, they put some additional innovations. Factor VIII gets activated by the molecule called thrombin, and they put in some recognition sites for the thrombin molecule so that when clotting is activated, thrombin comes in and it cleaves or snips the protein where those recognition sites are. And after it snips, you're left with this molecule here, which is the activated form of FC fusion factor VIII or a lactate. So this looks exactly like what a lactate would look like when it gets activated. So it's a pretty innovative molecule. Well, how does it perform? So here is a traditional standard half-life factor VIII with a half-life of about 11 hours. So you can see after infusion, it's pretty much down to baseline levels, certainly by 72 hours, so 3 days. What about an extended half-life molecule? There's a modest change in the half-life, and now it takes about 5 days to get down to sort of less than 1% levels. But what about the what's now called a fanosoctocog, or the BIV01 molecule? What you can see is that it stays in circulation at a very good level for multiple days, for at least half the week after dosing, this is in what's considered the non-hemophilic range. So with a single dose of this factor VIII, the factor VIII levels observed in the plasma are above 40%. If it's carried out to 7 days, patients still have levels of roughly 10%. And if it was allowed to wash out completely, it would probably take a couple of weeks to clear from circulation. So the way they've designed this trial is they are putting patients on once a week dosing of this efanesoctocog, and they are observing that patients are spending at least half the week where their factor VIII is in the non-hemophilic range. So this is potentially a new level of protection from a traditional replacement therapy, but with some really significant innovations added to them. Now, what I'm going to talk to you about next is this category of cross-platform therapies, and they all share these similar characteristics. So they're only used for prophylaxis. So you can't treat an acute bleed for these. You have to take these therapies and rely on them for your prophylaxis. If you have a breakthrough bleed, you're still going to need a traditional therapy. Factor if you have hemophilia A or B without an inhibitor, or a bypassing agent if you have an inhibitor. They are effective in both hemophilia A and B. They can be used in people with and without inhibitors, so this is going to increase access for a broad category of patients. There's no peaks and troughs between the doses. Once they're at steady state, they give even steady state correction of blood clotting. So far, all of them are amenable to subcutaneous dosing, so no more reliance on IV infusions. And overall, they have a relatively low burden of administration. Most of them can be given either weekly, as much as every month, or even every other month, which I'll show you. Now, here's the principle of what we have called this category, the rebalancing therapies. So what this is showing is it's thinking about blood clotting as a balance between the procoagulants, so the things that are trying to drive blood clotting, and then the natural anticoagulants of plasma, which are trying to contain or counter the procoagulants. Now, if we have a normal physiology, we have equal amounts of the procoagulants and the natural anticoagulants. That allows us to generate as much thrombin as we need to, but not too much, and enough to convert fibrinogen into fibrin, which is the stable blood clot that we need. So that's the normal state. Well, what happens when you have hemophilia, if you're missing factor VIII or factor IX? Well, now you've tilted the balance. You don't have the full balance of the procoagulants anymore, but all of the natural anticoagulants are there in full force. And so they are counteracting the remaining procoagulants. And now the balance is tilted to being anticoagulated or a bleeding phenotype. You can't generate thrombin anymore. And so you can't convert fibrinogen to fibrin. We can rebalance if we add factor VIII or factor IX here. But the real epiphany for me in thinking about this is you can also rebalance without adding any factor VIII or factor IX. If you simply target these natural anticoagulants, you can bring hemostatic balance back to a patient. So that's what we're going to talk about. Now, the concept for this is actually grounded in natural observations we've had in patients, okay? So there are some inherited conditions where patients have severe hemophilia, but they have behaved as if they had a very mild clinical bleeding phenotype. And it turns out, this happens rarely, but patients have been described where they have deficiencies of those natural anticoagulants, deficiencies of protein C, protein S, or antithrombin, or this molecule called TFPI. And so they've naturally rebalanced their hemostasis. Now, rebalanced hemostasis, we also see in liver disease. So remember, the liver is responsible for a number of production of clotting factors. And if you have liver disease, you aren't making those procoagulants anymore. And so you would think that that would be tilting you towards bleeding. but it's also balanced because there's a number of other factors that are either defective or are altered in liver disease that counterbalances the loss of those clotting factors. So in general, patients with liver disease generally have a balanced hemostasis. They're neither bleeding too much or clotting too much in most situations. So we're going to look at these three targets: tissue factor pathway inhibitor, antithrombin, and activated protein C. These are the three main regulators of coagulation. The coagulation system is trying to generate thrombin so it can convert fibrinogen to fibrin and make a stable blood clot. Once you're missing factor 8 or factor 9, this can't happen effectively anymore because of the full effect or the full force of these natural anticoagulants. So if we could target the TFPI, target the antithrombin, or target activated protein C, we could bring balance back to the blood clotting system. So let's look at these individually. So first, targeting TFPI. So these are anti-TFPI therapies. TFPI is what's called a protease inhibitor, which means it counteracts clotting by going after the proteases of coagulation. So these are the procoagulants. And it particularly targets factor 10A and factor 7A. And by doing so, it impacts thrombin generation. There are targets that have been developed that can impact the function of TFPI and basically inhibit it. And by doing so, it will rebalance coagulation. Some of the characteristics of these moles that they can be administered subcutaneously, so they're bioavailable. You give it sub-Q and they get absorbed readily into the circulation. They have stable pharmacokinetics, so you get nice, even hemostatic protection. And because we're not countering factor 8 or factor 9 or having to worry about the inhibitors, they work in all of those situations equally. Now, there are some challenges. We don't have a good way to measure these. So as we knock down the TFPI function, we don't have easy assays to measure and be precise about that knockdown. Low TFPI levels have been associated with increased clotting risk. But keep in mind, these are in individuals who have normal procoagulants. They have normal factor 8 and factor 9. If you have hemophilia, we think that there's a safety window there in that rebalancing. The other important part is that these are going to appear potentially as foreign molecules to the immune system, and there is the potential to develop anti-drug antibodies directed against these agents. This is one such molecule called marstacimab. So this is a TFPI inhibitor. These patients came into this trial, they were not on prophylaxis. So this was their numbers of breakthrough of bleeding events when they were just being treated on demand. But after they started on the marstacimab prophylaxis, look what kind of effective control. Most of the patients actually experienced 0 bleeds after starting on this prophylactic therapy. So this concept really does work. From the study that's been reported so far, there were no serious adverse events from this. Importantly, nobody had any thrombotic complications. And overall, we had a significant reduction in the annualized bleed rate, whether they had hemophilia A or B with or without inhibitors. So there's more to be learned from the clinical trials. This is still advancing, but I think this is a concept of therapy that could be one of these new non-factor therapies that we could have as an option in the clinics. Antithrombin's another target. If normally antithrombin is basically acting to dampen down the procoagulant action of factor 10A and thrombin. So if we could inhibit or knock down the antithrombin levels, we would be able to restore the thrombin generation, or the thrombin burst, we call it. So the molecule that does that the best so far is an interesting molecule. It's called a small interfering RNA. And this is given as a sub-Q injection. And what it does is it targets to the liver where antithrombin is made. And that RNA finds the messenger RNA for antithrombin, and it forms a complex to neutralize the message so it doesn't make any protein. So sometime after you receive this injection, your antithrombin level secretion goes down, and you end up with a very low antithrombin level in plasma. It has a sustained effect, and it will stay down at that level as long as you keep dosing with this drug called fitusiran. And again, it is effective in A, B, with or without inhibitors. Now, similar challenges we talked about. If you have antithrombin deficiency naturally, you are at increased risk of blood clotting. But again, this is in patients who have normal levels of all the other clotting factors. When you have hemophilia A or B, this gives you a potential safety window so you can rebalance hemostasis as you knock down the antithrombin levels. There have been some other interesting things. We think it's because this siRNA targets the liver. These are liver enzymes that we can pick up. These are asymptomatic elevations of the liver enzymes. We don't totally understand why it happens with this molecule, but it doesn't appear to be associated with any adverse consequences. Well, we just got a readout of the late phase trials, the so-called phase 3 trials, and they looked at this molecule in 3 groups of individuals: adolescents and adults with Hem A or B without inhibitors, that's Atlas AB; adolescents and adults with hemophilia A or B with inhibitors, that's Atlas INH. And then they looked at patients, whether they had A, B with or without inhibitors, who were on some form of prophylaxis, whether it be clotting factor concentrates or bypassing agents like Nova-7 or FIBA. And then they were switched to the fitusiran, and then they were able to compare the effectiveness. So this was also presented this summer. So this shows you what happens after you start taking fitusiran injections. Here is your starting antithrombin level, so we'll call that 100%. And within about 2 weeks, your levels fall down to very low levels, about 20% or lower, maybe as low as 10% of normal. And then it just stays down there as long as you keep taking the dosing. Accordingly, we have an improvement in the thrombin generation. So this is just simply by counteracting the antithrombin, the thrombin potential, or the amount of thrombin that the plasma can generate in this assay, is improved. How improved? Well, what's not shown on the graph here, but if normal individuals were also studied in this for a comparison, and normal individuals start to overlap here in the upper range. So fitusiran seems to be taking us to potentially a new tier of correction that we haven't been able to get with some of the non-factor therapies. How well did it work? Well, in the Atlas AB, the non-inhibitor patients, compared to on-demand therapy, there was a 90% protection from bleeding. In the Atlas inhibitor, compared to on-demand bypassing agents, again, over 90% protection from bleeding on fitusiran prophylaxis. But the big punchline comes from the prophylaxis comparison. So remember, these were patients who were either on factor or bypassing agent prophylaxis. These were the annualized bleeds that they were having on this therapy. So about 4 or so bleeds per year, and a minimum proportion of the patients experienced 0 bleeds while they were on this prophylaxis, only about 11%. After they switched to fitusiran prophylaxis, their annualized bleed rates went essentially to 0, and 41 of the individuals, or 63% of the individuals, had 0 bleeds while on fitusiran prophylaxis. So the last one is the newest kid on the block. This is an activated protein C agent. Same purpose. Activated protein C dampens down the procoagulants because it targets factor 5 and factor 8. And this limits the amount of thrombin that can be generated. So if we can knock APC down, we can restore thrombin burst and have efficient blood clotting. So what this agent does is it acts, I think the equivalent I would call it is it's kind of like a mousetrap. And so it identifies activated protein C and it clamps down on it and it inactivates it. And it has a very long half-life, about 5 days. So once weekly dosing or even longer is potential with this therapy. It can also be given subcutaneous, and it can also be used in patients with hemophilia A or B with or without inhibitors. Now, same thing as we talked about before. Well, doesn't protein C deficiency, isn't that associated with blood clotting? It is, but that's in patients who have normal levels of factor VIII or factor IX. We've got a buffer here, a safety window where we can knock down APC when we have very low levels of factor VIII or factor IX. I don't have any data on this one. The first reports are going to be come out at the hematology conference in December. So these therapies overall are going to potentially reduce the treatment burden for hemophilia because they can actually be given fairly infrequently, and they can also be given subcutaneously. So they're all subcutaneous route of administration, but look at their frequency. The AT lowering agent, fitusiran, can be given once monthly or actually once every month. So now you're talking about 6 subcutaneous injections to provide effective prophylaxis in the course of a year. The anti-TFPIs range from a daily sub-Q injection to a weekly injection, and the anti-APCs are probably going to be weekly to once monthly. The nice thing about these agents, just to remind you, is because they look nothing like a factor VIII or factor IX molecule, they can't be neutralized by inhibitor antibodies. So they're going to be effective in patients whether they have had a history of inhibitor or have an active inhibitor or not. Most of them do not induce an immune response, but they could. So that when we see that, we call these anti-drug antibodies. These are being evaluated in the clinical trials. We don't know the frequency yet, but they're probably relatively infrequent. But the beauty of these agents is they can be used for all comers, A, B, with or without inhibitors. And the thing that I get excited about is thinking about how do we improve access to the world. We've had clotting factor agents, clotting factor concentrates since the early 1970s, and still in 2022, 75% of the world has no treatment products available, certainly available for prophylaxis. So could these non-factor therapies be less resource intensive and less cost prohibitive, so we could actually get these and use them in the developing world? The World Federation, is really working to get rid of treatment disparities across the world, and many of them are looking at these new therapies, hoping that they can help overcome some of the challenges we have in the developing world. And of course, these are less burdensome. So when you think of some of the logistical challenges of treating patients in Africa or Indonesia and some of these other places where WFH is working with humanitarian aid, etc., having access to non-factor therapies could be a real boon to improving access for patients. So I'm really hopeful for the future with these therapies. None of those latter ones I've talked to you about today have been approved yet, but some of them I think are very close. They've wrapped up their phase 3 trials. They're going to be submitted to the FDA and the European authorities for review, and we hope that we'll have at least some of these available for offering patients as an option. So let me see if I can address some of your questions. Oh, talking about health equity. There's no reason these therapies can't be used in women with bleeding disorders, and including women who have hemophilia. There is even the potential that these therapies might have some function in rare bleeding disorders. So again, looking to these therapies to broadening out the health equity. What have you heard about bone density issues with the sub-Q option? So I understand what you're asking. It probably has nothing to do with the sub-Q administration. The principle that we're looking at here is factor replacement, and particularly factor VIII replacement, is associated with some functions that go beyond blood clotting and also function at the level of bone resorption and mineralization. And so people have raised concerns about, well, if we don't give factor VIII back, does that mean that patients will have sort of low bone density? Well, if that was true, that would already be the case for patients with inhibitors. And the only reason we see any sort of alterations in bone density issues in inhibitor patients is because of bleeding, not because of missing out on factor VIII. The other thing is most of the mechanisms that have been described, if not all of them, are related to the ability to generate thrombin at the level of the bone interface. And all of the therapies that I talked to you about are working to enhance thrombin production. So I don't think any of these are going to end up proving that they are shortchanging anyone with regards to bone health. Let's see, from Will. When some of these novel treatments come out of the market, what will a comp care visit look like? What sort of questions will you discuss with patients? Yeah, so I guess we're getting the first experience with that, Will, with emicizumab. So what do we talk about with our emicizumab patients? We want to make sure they're, staying, adherent to the prescribed therapy. The beauty of emicizumab, though, is because it has such a long half-life. If patients miss a dose here or there, it really doesn't have the same implications. If you're on a standard half-life factor VIII molecule and you miss a dose, you're not protected anymore, and your level's right down in your boots. And so the risk for breakthrough is quite high, but it takes months for you to fall down from steady state levels if you stop emicizumab completely. And that's true also. So I'm most familiar with fituzaran because we had several patients on that in the trials. And when we washed them out from the fituzaran dosing, it took about 3 to 5 months for their antithrombin levels to go back up to the normal range. So the consequences of adherence are less. I think we'll be spending a lot more time talking about how are they managing breakthrough bleeding, because all of these therapies I showed you involve having to make some modifications to how you dose for breakthrough bleeding events. When you rebalance your hemostasis, you're at a new level of protection. So you can't just treat willy-nilly with your normal factor VIII or factor IX doses or your bypassing agents. You risk actually overshooting on the thrombin correction. And so because of that, all of these therapies do run the risk of breakthrough thrombosis. And most of the trials have had thrombosis events occur. Almost all the events occur coincident with treating for a breakthrough bleed. So most of the programs have a risk mitigation where they have explicit instructions on dialing down the doses for managing breakthrough bleeding in order to minimize that overshoot. So I think that's going to be a key part of the interaction in comp clinic, is making sure that patients understand, if you have a breakthrough bleed, what's your dose? How frequently can you give it? Making sure that they know about this potential risk. Are these newer products FDA approved yet? So the answer is no. Emicizumab is the first in the category. If it's any example of the impact of what these therapies can be, emicizumab is the #1 prescribed prophylactic agent for patients with hemophilia A. But doesn't work for Hem B patients. At least it's not approved for that. And we don't have a non-factor therapy for patients with hemophilia B. All the therapies I talked to you about tonight have some potential for patients with hemophilia B. So I would love to get us to the point where at least one of these products were approved and we had a sub-Q option that has the low treatment intensity, low burden like emicizumab does, and be able to offer that for Hem B patients, and particularly to have an option for our Hem B with inhibitor patients who are really challenged by their underlying condition. Anyone else have any other questions? Those were great questions.
So Dr. Pipe, there was one more that came in. It says, can mild females be subject to throwing blood clots, and how is this balanced?
So I mean, at a fundamental level, I can say anybody can develop a blood clot, depending on what's going on in your physiology. And even patients with severe hemophilia have had blood clots. So there's probably nothing unique to either being female with a bleeding disorder or to having a non-severe hemophilia. What would happen if you use these agents in a non-severe scenario, if that's maybe what the person was asking about. Right now, these are only being investigated in severe or moderately severe forms of the disease. But if you think about the new tier of correction that I talked to you about, there's no reason that these agents wouldn't rebalance you if you had a mild phenotype as well. I would hope that if they'll get approved, hopefully, for the severe patients first. But then I think we'll have to learn, is it safe to use these products maybe at a lower dosing range to help rebalance patients who have mild hemophilia and really give them a lifestyle that's indistinguishable from their unaffected peers? So if that's what the individual is asking, I think I'm actually pretty intrigued by that prospect. We don't want to have therapies that are only available for patients with severe forms of the disease. We want to make sure that these can be available for milder elements as well.
I think that answered all the questions that came through while you were presenting. So if anybody has any others, we probably have a minute and we'll move on. But thank you so much for that presentation. It was really clear and it sounds like there's some really exciting options coming for patients with bleeding disorders. So appreciate your time and your expertise tonight. Thank you.